Hi everyone, this is the last lecture for chapter 19. This is on DNA repair. So we've been talking about different mutations that can happen in DNA, but probably you've realized that for the most part, we are still managing to survive. All these organisms are managing to survive, which means that many of these mutations must be able to be repaired. And so we're going to be looking at some different types of DNA repair mechanisms, and then talk about how some specialized DNA polymerases can actually synthesize DNA over a damaged region and actually fix that damaged region. We'll talk about a bunch of different types that we would see within this. So DNA repair has to happen because most mutations, now not all, but most are what we would call deleterious, which means that they actually can be lethal or make it so that the organism is not going to at least reproduce. So these DNA repair systems are vital to the survival of all organisms. And so all living cells contain several different DNA repair systems that can fix different types of DNA changes. In most of these cases, though, you need to think about it as a multi-step process. It's not just that it comes in and immediately fixes. In general, what happens is kind of a three-step process. The irregular DNA structure is detected, so whether it's a frame shift or it's a deletion or it's a base insertion or something else, a thymine dimer, first it has to be detected, recognized. When it's been recognized, generally what we see happening is that abnormal DNA is then removed, and then we have the synthesis of new DNA, the normal DNA, to replace what had been changed. We have some different types, and this summarizes them. We have our direct repair, and that means an enzyme just recognizes that there's an incorrect alteration and converts it back to what it should be. This is, generally speaking, a small DNA maybe mutation. Maybe it's just one base mispairing or something. We can also get base excision repair and nucleotide excision repair. This is also like above, that if there's an abnormal base or nucleotide, it can be removed and then that segment can be excised, and then that new complementary strand is used to build a new normal DNA strand. We can also get mismatch repair. So similar to that excision repair, the DNA defect is a pair mismatch and not an abnormal nucleotide. So maybe it's an A to a G. This mismatch then would be recognized, and then the parental strand is used as the template to synthesize that normal daughter strand. So you still uses the parent strand, which hopefully is not where we saw the mutation. Homologous recombination repair. If there's a double strand break in the DNA, often what can happen is that the strands of the normal sister chromatid can be used as a template to repair that damaged sister chromatid. And then the last one, the non-homologous end joining. This occurs if we have a double strand break as well, and the broken ends can be recognized by proteins that actually can join them back together. And we'll go through each of these in this particular part of the chapter. So direct repair, in a few cases, that covalent modifications of nucleotides can be reversed by a specific enzyme. So, for example, if we have that dimer that I talked about with UV light, there's a particular enzyme called photolyase that can come along and repair that. It will actually split up the dimers and return the DNA to its original condition, and it uses light, so it's photoreactive in order to work to get this catalyzed. There can also be alkyl transferase, which repairs these alkylated bases, and it can transfer the methyl or ethyl group from a base to a cysteine side chain within that protein, and then what this can do is permanently inactivate any alkyl transferase. And I think I have a example of that. So first of all, here's our thymine dimer, right? Remember, these would normally be paired with adenine across from it, but if there's UV light damage, they can actually form a dimer between the two thymine. This DNA photolyase can break this bond and allow them to go back to their normal structure. So that's that direct repair with a thymine dimer. We can also get the direct repair of a methylated base. So in this case, we have a methylated methylguanine. We have our alkyl transferase that can come in. It can actually remove that methyl from this and completely take it away, and we go back to what would be our normal structure of guanine. I also talked about base excision repair. And in this case, it involves a category of enzymes known as DNA N-glycosylases. And these enzymes can recognize an abnormal base and then break that bond between it and the sugar in the DNA. And so this particular base then can actually get that started, and then we have something called an AP endonuclease that makes a cut on the 5' side. Depending on the species, it can then eliminate the abnormal bases, so perhaps a uracil, so if it has put in a uracil or a thymine dimers, but it can also do 3-methyladenine or 7-methylguanine, and I think I have a picture of that. So in this case, we have a uracil that was incorrectly base paired here, and N-glycosylase will come in and recognize that that base is not right, that it's an abnormal base, and it will actually remove the base from the sugar. Then we have this open spot here, and AP endonuclease will recognize that and actually cleave that DNA on the 5' side. What's going to happen then next is that we are going to be able to replace that. And so in E. coli, we have a polymerase that can use that 5' to 3' exonuclease activity, right? Removes that, and that happens in the bacteria, and then it fills it with the normal DNA. It's a little bit different in eukaryotes. In eukaryotes, it's going to be the DNA polymerase beta that can remove that apyramidal site and replace it with a correct nucleotide. So it removes it and replaces it. And in eukaryotes, such as humans, we can also have DNA polymerase gamma or epsilon that can synthesize a short segment of DNA, which generates a flap. And when that happens, it can be removed by the endonuclease, and that area gets sealed. So in any case, that where that uracil originally was will be replaced and brought back to where it should be. We can also get nucleotide excision repair, and this can repair a lot of different types of DNA damage. It can replace those thymine dimers and chemically modified bases. It can also help if there's a missing base or some type of cross-link. So these nucleotide excision repair systems are found in all eukaryotes and prokaryotes, but of course it's most best understood in prokaryotes and most studied in prokaryotes. So in E. coli, this nucleotide excision repair system requires some key proteins, and these ones are UVRA, UVRB, UVRC, and UVRD, and they're ultraviolet light repair of these pyrimidine dimers. They are also really important in repairing chemically damaged DNA because they can recognize and remove short segments of damaged DNA. So once they've removed that, what you can have is you can have DNA polymerase come through and then add back in the correct bases, and ligase will stick those to the ends where that replacement took place. And so this is showing you these are the different proteins. They come in, they're kind of reading this DNA, searching for any damaged DNA. When they come across this thymine dimer, they recognize there's a problem there. They actually will then bind, the last two proteins will bind into, the last protein UVRC will bind in, and they will actually cut out on both the 5' and 3' end that basically bookend that thymine dimer. When they cut that, they actually remove then that whole section. So it removes the whole entire damaged region that bookends this thymine dimer, and then the UVRC and B are released, and we have this opening here. DNA polymerase can come along and build that back up so that we fill the gap here with no thymine dimer, and ligase will stick it to both the 5' and 3' end, replacing that whole part. We can also get mismatch repair, and so in this case, where we know that the structure of DNA should be following the AT and GC rule of base pairing, we know, though, that during DNA replication, it doesn't happen very often, but sometimes an incorrect base might be added, and this would create that base pair mismatch. So DNA polymerases have a 3' to 5' proofreading ability. They basically run the length of the DNA looking for any mismatches, and then what they do is they try to fix them. If it gets missed, though, by this DNA polymerase, we actually have a proofreading we have a backup to the proofreading, and that's the mismatch repair system. And this is found in all species, and in humans, mutations in the system are one of the places that we see associated with some types of cancer. So it's a problem that we could have that mismatch. So mismatch pairs, again, have been studied extensively in bacteria. And in this case, we have these proteins, these enzymes that can detect the mismatch and direct the removal of it from the newly made strand. So this is on the daughter strand. The cool thing about these proteins is that they actually can distinguish between the parental strand and the daughter strand. So you can imagine if there's a mismatch, it's important to know which is the correct bases to use as the template. And so what ends up happening is that the mutH can actually distinguish, because prior to replication, both strands are methylated. Both of those strands are, and so the parent strands are replicated. And immediately after the replication, what we know then is that the parental strand is methylated, which allows it to be recognized, while the daughter strand is not. So this gives a quick picture of this. We have our parental strand here, right? It's the dark purple in the background. Then we have this light purple that's the inside one, which is our daughter strand. And what we know is that we have a methylation. We have that methylation on our parental strand, which allows these particular enzymes to say, okay, then we want to use the parental strand as the template. And so what these will do is they'll come along and bind to the daughter strand, and they'll actually remove the area where there's the mismatches. And they know to remove it from the daughter strand because we have the methylated parental strand. And now we have this open patch that needs to be fixed. And what we can see is that we will have an actual DNA polymerase that will fill that vacant region, and then DNA ligase will seal the end. But it's being able to have that methylated parental strand that allows us to know which strand to use as our particular template. We can also get double break strands in DNA, and these are very dangerous. If you get breakage of a chromosome into pieces, what we can imagine is that this can cause chromosomal rearrangements or even deficiencies if it breaks down. And this is often caused by ionizing radiation, sometimes by chemical mutagens or free radicals. And we get about 10 to 100 breaks occur each day in a typical human cell. So they have two systems that can repair it. We have our homologous recombination repair or our non-homologous enjoining. So with homologous recombination repair, the really cool thing is that with our sister chromatids in this point, we have identical DNA strands that are side by side. So if we have a double break strand, what we can do is basically trim those so we get these overhanging ends to our damaged strand. And what we'll get is our sister chromatid will actually kind of invade there and allow it to be used as a template. And so what ends up happening is that this purple strand that was not damaged can pop open and actually allow itself to be used as a template, so that now this one that had been broken has something to create a new strand that's complementary to its sister chromatid, and you end up having that resolution and ligation with the correct strands. Now we could also get what is called non-homologous end joining. So this, we have breaks of both ends here. And in this case, what we can get is we can get these end binding proteins that will actually bind. We'll get a protein cross bridge, and then we'll have all the proteins will come in for DNA crossing, and it will fill the gap and ligase. Now in this case, we don't see necessarily that we get exactly what the original strand was, but it actually keeps the chromosome as a whole. And so what we see is that with this one, you may not get high fidelity, but at least you won't have part of the chromosome lost. We do have translesion DNA polymerases. These are when we have lesions that have escaped any of the other DNA repair systems, and typical DNA polymerases cannot replicate through a region of DNA damage. These have the ability to fix this, but they have a very low fidelity, which means they're very error prone. So for your homework on this particular one or a question, you should discuss the similarities and differences between the nucleotide excision repair and mismatch repair systems. And that's the end of Chapter 19. Chapter 20 is going to be on some molecular biology lab techniques. If you have any questions, let me know.